Welcome back, everyone, to the OIS Retina podcast. This is Firas Rahhal, the host, partner at Retina Vitreous Associates in Los Angeles, and a partner and founder at Excite Ventures, centered in New York City. I'm overwhelmingly delighted and happy to have this guest today. This is a longtime personal friend of mine. Most everyone in the ophthalmology, certainly retina space, knows him and probably knows him personally. He's a super successful guy and also a super social, fun guy. Ram Palanki, my 15 to 20-year friend at least. I lost count somewhere. Ram is currently the Executive Vice President of Commercial Strategy and Operations for REGENXBIO. We're going to talk a lot about that in a few minutes. First of all, Ram, welcome to the program and thank you for joining us.
Thank you, Firas. And it's always good to chat with you, my friend, on podcast or at a dinner table or during a conference. So really glad to be here with you.
Thank you. Yeah, truth be told, Ram and I dine out together a lot. We both love food and love fine restaurants. So we've had a lot of time to talk science and personal life and enjoy each other's company. So I should give full transparency on that. But he's here professionally today. He's the best in the business. I'm going to go through some of his historical positions recently and then talk to him maybe about one or two of them to get some background. So previous positions for Ram include Senior Vice President of Commercial for the Americas for Santen, Global Head of Marketing and Sales for Thrombogenics, Director of Global Marketing and Sales for NeoVista. You're all getting the picture. This guy is a master of product development in the back of the eye and of marketing and commercializing those products. We're going to spend a lot of time talking about those skills and those issues today. But I have known Ram longer than that, since his days at Genentech, now Roche Genentech, when he was one of the integral members of the first team that launched Lucentis, a very important part of our retina history in the last 20 years. It turned out, and I didn't realize this until I reviewed your record, you were also part of the Macugen launch, which even predates that. I'm not sure you and I had even met at that time. I was in New York in those days. What can you tell us about that crazy, exciting, super successful time with two great products coming online for diseases we'd never even dreamed of treating? What was that like?
I mean, for us, it's, I think about my journey starting with Macugen to Lucentis to what I'm doing today at REGENXBIO, right? So, and I go back and think about historically the contributions of Macugen and Lucentis and the evolution from there to Eylea, Beovu, faricimab now being in the marketplace, and all of the innovation that's happening to solve for the unmet need that Lucentis has created after its launch in 2006. If you go back and think about Macugen days, I look at Macugen as the starting point for innovation in retina. Pre-Macugen, this was predominantly a surgical field with less than 1,000 specialists, and the conversation they could have was really to tell patients that they could probably slow down the progression of the disease with a surgical procedure for a few months to a year, but they're ultimately going blind, right? I think what Macugen brought forward was actually an in-office approach to treating macular degeneration. Of course, it wasn't as good as what Lucentis could contribute, but it was a starting point. I think Lucentis came into existence because Macugen was being explored as a potential anti-angiogenic. The approach with Macugen was a isoform of VEGF, which was preclinically shown to be predominantly involved in the angiogenic process, which is VEGF 165. And I think Lucentis took the approach of blocking all isoforms of VEGF-A, and that was based on a lot of the oncology science that was evolving at Genentech with Avastin and such. So I think those contributions were all based on sound science, and in the clinic, it was clearly shown that blocking all isoforms of VEGF-A was really critical, and Lucentis really, brought forward the innovation that Macugen had started to a next level of contribution on the basis of science, where the product was named to be the top 10 discoveries in science during 2006 across all fields of science. And I think that has changed retina forever, right? It went from a specialty of 800 specialists that was predominantly surgical to a specialty of over 2,100 specialists now, which is predominantly surgeons that are performing medical procedures day in, day out. I think that evolution has been a process that I've observed how when a treatment really works, that infrastructures as well as operationally what's required to take care of such a large public health priority gets adopted when the right products are introduced to market in a disruptive way.
Yeah, that's totally correct. And I didn't know the numbers about the number of retina doctors, and it changed. I was obviously in practice in those days since the mid-90s for me, and I knew we were fairly boutique, and we still sort of are by comparison to other specialties. But that's almost a tripling of the doctors, and it speaks to the number of patients we are now able to treat. And that's about to continue to explode more, as you know. Did you know then, Ram, you're an intuitive guy, you're a smart guy, you were earlier in your career, how landmark those moments were?
It's when I came from an oncology background, Firas. My upbringing was at Novartis Oncology. I spent my early days doing my fellowship and spending my time as a young professional, looking at financial models and assumptions around NPVs that would contribute to making decisions on how you move things from early stage to late stage and look for business development opportunities. When I first came across the Macugen team and the iTech team in their early days, having seen what was happening with the science of angiogenesis in oncology at that stage, I knew this science had a big role to play in ophthalmology. How big was it going to be? I only realized that after the Lucentis launch, in many ways, coming into that organization. And, for us to, when I was there, I came in 2 years before Lucentis phase 3s even read out. I was one of the first 3 people at Genentech working on the ophthalmology franchise. A little bit of history for you. And that actually gave me an opportunity to work across all functions of drug development, from clinical, medical through commercial. And as part of the process, when we started thinking about the introduction of a product that needs to be administered for your lifetime and with no sign of diagnostics that could actually tell you how the drug was working, it wasn't even something you could imagine. I mean, these are all the things we were thinking about. If you go back and look at the concept of OCT in early 2000s, it didn't exist, right? I mean, there were no OCTs in Marina and Anchor. There were maybe 30 patients towards the end of the Anchor trial that had OCTs. And if you look at those images, you don't even know what's going on with those images, really. But it's the introduction of OCTs and the contribution of that to anti-VEGF therapy and the concept of this lifetime treatment, all these things, we had conceptually imagined would have a huge impact, but the type of impact it has had now, coming into 2022, you couldn't imagine that this would be a mainstream treatment where patients are getting treated for their lifetime, and that OCT would drive every decision you make in the clinic. We had some view into it. I mean, I was involved with the Pronto study, if you remember, the study that was that came out of Bascom Palmer with Phil Rosenfeld. I mean, me and Phil partnered on the protocol. We didn't know what would be a good retreatment criteria. We talked about 150 microns to be a retreatment criteria that kind of went to 100 microns to 75 to 50 microns. So that process of evolution and having lived that entire process of thinking of introduction of diagnostics through how anti-VEGF therapies can be managed on a sustainability basis, those were concepts we had. And in some ways, we were forward-looking in terms of working with people like Phil and Bascom Palmer and Carmen back then, and even Jay Duker and such, that were all involved in the diagnostic evolution of OCT, to in fact do some of this work even before Lucentis got introduced to the market. And I think that really helped to pave way for a sustainable way to treat these patients, given the number of the ratio between providers and patients just never really could keep up with the sustainability concept. And even today, you're struggling, right? It went from 800 clinicians to this, over 2,000 physicians, but you're consistently struggling with treating patients in a sustainable fashion and being able to treat your patient load across different disease states that you manage, from diabetic disease through now dry AMD is coming into view. So this whole evolution for me has been a process of learning and also contribution, where we were thinking about a lot of forward-looking strategies in terms of introducing things that would help clinicians manage patients in the best way possible.
Yeah, I'm glad you brought up the OCT. We often forget this part of it, especially people who came into the field after. It was brand new then, and I remember early 2000s practicing without one very effectively, but we couldn't have done all this 40, 50 injections in a day, seeing another 40 patients on top of that, without the speed of information gathering that the OCT gives us. The other thing that strikes me about that time, and it speaks a little bit to what you were saying, I don't think I knew then how game-changing it was going to turn out, but I do remember one thing, and Dave Boyer and I have talked about this a lot. We were in the trials, as you know, Marina and Anchor and all the trials. It was the first time in our kind of clinical trial experience, and there have been maybe others after, but really that was the principal time that the masked examining physician was almost certain who was getting treated. There was like no doubt in the mind of the masked physician. It was almost embarrassing to be masked because it was the first time we ever saw any of these people actually get better. It was remarkable and exciting. And we knew it was super important. It was RuFab at that time, as you may recall. And we used to talk about it like, hey, it's too easy to tell who's getting treated, right? Not by unmasking yourself, just by seeing what the macula looked like.
And I used to hear that all the time when I was talking to clinicians that were participating in the phase 3 programs. Genentech's relationship was predominantly in the oncology space and immunology space. And for me, having been part of that very small team, I was almost forced to get out there and get to know all of you. And in some ways, that opportunity that Genentech gave me and the contribution of Lucentis as science put me really in the middle of my professional evolution. And that core development is who I am today, having interacted with all of you on such a consistent basis at all of the major institutions through private practices, learning what drives you as a retina specialist, as a surgeon, as someone that cares immensely about your patients, and how you think about patient care and where you were back then to as Lucentis got introduced and the evolution of Avastin through the process, and the contribution of both of those drugs in the context of science through phase 3 programs, through anecdotal data, and how all of that got adopted into the marketplace with one single intent, right? To bring the best product to the patients from both sides of the equation, from a corporate standpoint and from a clinical community standpoint. And here we are. We have introduced so many products through the process. And again, the process continues to evolve. And for me, in many ways, having launched Lucentis and now coming to solve the problem that Lucentis had created as an introduction to the marketplace, right? So Lucentis solved the problem of blindness, but introduced the problem of sustainability. And that really has translated into blindness for patients over the long term, right? Because nobody is able to sustain that chronic frequent treatment with these injections. And hence, over time, over and over, the real world data shows that patients ultimately lose vision and go back to baseline. And for me, being at REGENXBIO, trying to solve for that problem with a one-time gene therapy has really brought me the full circle of what I was part of with Lucentis in that early 2000s to now being in 2022 and solving for that issue.
I'm so glad you went to this topic, because my next topic really is the current situation and where you are now. And I want to give you every opportunity to talk about that. And we want to hear about it because this is super exciting stuff. As I mentioned earlier offline, I just lectured a little bit on this, and I find the REGENXBIO story and the gene therapy story super exciting to talk about and to present to laypersons, other people in the eye business who don't know the retina space perhaps, and that you see their excitement and engagement when they hear about these modalities. So just in review, again, your current position, Executive Vice President at REGENXBIO, you're in clinical trials. I've always been a fan of this idea of the biofactory, which is essentially what's being done here. Others are trying it as well. Maybe you could speak a little bit, because you're the expert on this topic, to our viewers: mechanism of action, the delivery method, the vector, on some 20,000 foot level. What's the story? What are you doing here? And how does it work?
Maybe I'll introduce some of the historical concepts with gene therapy too here for us, so we can step into what we are trying to solve for with RGX-314, which is our lead product candidate that's in phase 3 program today, based on the data that came into view a couple of years ago in terms of the concept of one-time therapy. And if you step back and look at gene therapy itself, REGENXBIO has been at the core of its introduction and evolution in many ways, going back about 12 years into the history of the company. Our CEO, Ken Mills, who founded the company, really went out to seek gene therapies that were effective, not just safe. I think mostly what was happening in the gene therapy space was people were trying to take the availability they had to naturally occurring AAVs, and AAV2, most widely used institutional gene therapy, AAV-based serotype vector. And that has been consistently shown that it can be safe, but on an efficiency basis, AAV2 has the lowest efficiencies in the context of transduction and tropism to the right tissues. These are all concepts that are really important in gene therapy, and that's where Ken went out to seek discoveries that had gone beyond AAV2, and he met Jim Wilson at University of Pennsylvania, and that kind of translated into where Jim Wilson himself was trying to find naturally occurring AAV vectors that were effective and safe and had this selective propensity to transduce different tissues. And that process of discovery led to about 100 different serotypes that he was able to discover, including AAV7, AAV8, AAV9, RH10. There's a whole set of serotypes that belong to REGENXBIO as part of that discovery process that was licensed through the work that Jim Wilson did at University of Pennsylvania. That concept of tropism on a selective basis led to AAV8 being the most efficient vector in terms of transduction in the retina that could be safely administered into the back of the eye. So that translated into the evolution of this concept of vectorized antibodies, where we actually put a transgene into AAV8 that in fact puts the transgene into the retinal cells, and that gives the cells the ability to produce a therapeutic anti-VEGF Fab, which is very similar to Lucentis, over the concept being over lifetime. So that's the basis of the RGX-314 program. Most people think about gene therapy in the context of monogenic diseases, where you're trying to solve for mutated or genes that don't exist compared to wild type presentation of the human genome, right? So that concept has led to a lot of the original work, especially when it comes to retina in the IRD space, where most of these inherited retinal diseases could be solved for by putting back the correct transgene into the cells. But this concept of introducing a transgene that can encode for a therapeutic protein for your lifetime is a concept that we are exploring, for the first time in the context of wet AMD, DR, and 2 different routes of administration, subretinal and suprachoroidal. And to date, the data that we have observed and the maturity of the data that has come through the subretinal route has contributed to us moving the 314 program into the phase 3 program. So currently we're enrolling actively into 2 phase 3 programs. One of them is comparing it against every month Lucentis, and the other one is comparing it to labeled Eylea dosing against one single treatment of RGX-314 administered in the subretinal space in the OR. You're part of that program, so you know everything that's going on in that space. But that program was installed based on our phase 1/2 data that showed on a dose escalation basis, we identified a dose range that was showing us that on a minimum basis, we could probably accomplish about 50% of patients going injection-free on a long-term basis, and about 60 to 70% reduction in overall disease burden in terms of pre and post injections, post RGX-314 administration. And that trial was all done in patients that were previously treated and had long-standing disease for over 7 years, and that in some ways had a lot of recalcitrant and minimally responsive patients to anti-VEGF. And when we accounted for all of those, we saw a profile that we felt like we can move into phase 3, and that translated into the 2 phase 3 programs today, Atmosphere and Ascent. And you're part of those programs at your institution.
Yeah, they're brilliantly designed clinical trials, man. And I know you had a big hand in that, as did others, Steve Picola and others. They're really well-designed programs. Talk to me a little bit about the route of administration. I know that there's a suprachoroidal administration and a subretinal, and we're doing both of those trials, both of those techniques in our institution. Other companies have attempted intravitreal. There were some safety issues, at least in one of the arms of their trial with one of their indications. Without, no crystal balls here, obviously. Was this the thinking at REGENXBIO? Was it a safety concern to not go for intravitreal, or were you thinking the efficacy would have likely been better? And what are your thoughts about the 2 routes you're using in those regards?
I mean, this is all based on the science that exists in the public domain, Firas. I think we just talked about the serotypes. I think when you think about AAV gene therapy administered in vivo, you have to really think about what type of serotype you're using, because every serotype has a different efficiency in terms of transduction, as well as its ability to be able to dose through concepts such as neutralizing antibodies and the context of that neutralizing antibodies limiting or present some challenges when you administer the therapy into immune privileged versus non-immune privileged areas. So the data preclinically has consistently shown few components that AAV8 is way more efficient in transducing retina, and it is way more efficient in transducing retina. When I say AAV8 compared to other AAV serotypes like AAV2, modified versions of AAV2 that people use in the public domain today, it has been consistently shown on a head-to-head comparison that AAV8 is the most efficient serotype to be used to transduce the back of the eye. And then you think about what's the best way to do that. You have 3 options, right? You can either go the intravitreal route, which, once you put the vector in the vitreous chamber through the intravitreal route, you're exposing the vector broadly to the entire eye, including the anterior segment. And then you have to consider, in that context, what could be the risks, right? That could create potentially a risk related to inflammatory response through not being as efficient in transducing the back of the retina, but also the rest of the front of the retina, right? And you take those concepts and compare them to on a preclinical basis, the data has consistently shown that as you dose escalate, the intravitreal route is prone to be the most inflammatory route of administration. Hence, and then you step back and think about the subretinal route. Because the subretinal route is relatively immune privileged, it has been consistently shown that when you actually put AAV vectors into that space, be it any serotype, you're really not compromising the vector being promiscuous and transducing other areas of the eye, and it's really broadly transducing the intended tissue, which is really the retinal pigment epithelium, photoreceptors, and such. So that concept has been tested and over in not just in preclinical, but in clinical, even before us in other wet AMD trials, and has been consistently shown that that's the safest route to go. And I think by installing AAV8, what we're also showing is that we have a very efficient vector that can transduce these retinal cells, translating that on a dose escalation basis to therapeutic protein at doses that are very efficient, probably 100 times more efficient than other vectors. So the advantages there really become, with a smaller dose, you can get a broader transduction and higher therapeutic protein production in a immune privileged space where neutralizing antibodies don't matter, because in most other disease areas, not in ophthalmology, that's a big consideration. Your serotype you use and pre-existing immunity and the contribution of neutralizing antibodies on a efficacy basis becomes really critical.
Yeah, it's amazing how much has to go into this with regard to planning and understanding. With regard to the indications, you mentioned some, of course, diabetic retinopathy and of course wet AMD. With regard to diabetic retinopathy, as you know, there's like terrific level 1 clinical trial data that anti-VEGF given on some regular interval in advanced non-proliferative retinopathy without DME is a phenomenally compelling treatment paradigm. And I love it when I use it, but the fact is, as a field, we haven't adopted this modality into our practices or for our patients nearly as much as the robust data set would suggest. In fact, when I sit, if I have a patient who is super high-minded and can understand clinical trial outcomes with me, and I'll spend a few minutes and go over how compelling the data is, many of them will be like, hey, that sounds pretty good. I think I want that. You and I know, I think everyone knows that one of the reasons we've not been motivated is this idea of repeated injection into 20/20, not healthy, but say 20/20 or 20/30 eyes has been somewhat unpalatable for whatever reason to the retina community and or their patients. This seems to be a real opportunity for a longer term therapy, and I know you're studying this. What are your thoughts on this? Does this become a much more palatable way to treat well-sighted eyes with advanced non-proliferative diabetic retinopathy because of the durability?
Yeah, I think, we've always known prevention is the best cure, right? The problem then becomes, what type of assets and therapies do you have available that really substantiate the risk benefit on a preventive basis? I think what has been happening in the diabetic retinopathy space is, given the types of therapies that you have had access to, and given the type of evidence that has been generated in phase 3 programs with anti-VEGF, since Lucentis days to Eylea days, right, with Panorama. That was very exclusive to non-proliferative diabetic retinopathy patients, clearly shows that if you're able to treat patients with chronic anti-VEGF therapy, you can stop them from ever getting to those vision-threatening complications like DME and neovascular glaucoma and all of the other problems you have with proliferative diabetic retinopathy and such. And that's where, you're really trying to treat patients from going blind, but it's kind of late, right? But that's the best option you have today, given the disease progression and the risk benefit of these therapies. I think where we get really excited about the DR program is given the type of unmet need that exists in a public health priority, I mean, for us, there are 8 million patients in the US alone. And if you take the major geographies between US, the European 5 countries that we talk about a lot, UK, Spain, France, Italy, Germany, and Japan, there are literally 25 million patients today that are just sitting in the clinics and waiting for the disease to progress, which it will progress, right? I mean, once you have severe non-proliferative diabetic retinopathy, you literally are guaranteed to convert to DME and other vision-threatening complications like PDR and others in 50% of patients within a year. And if you have a milder, moderate disease, that might take a little bit more time, but everybody gets there eventually. But if you can stop them from ever getting there, I actually think you preserve the optimal vision and ability for these patients to be independent. And that's the concept we're trying to bring forward with our DR program, where with a single in-office administration into the suprachoroidal space, that we will hopefully prevent the progression of the disease over the long term and really put these patients back to their normal life. And they're notorious for non-compliance, and help US providers to provide best care in protecting their vision and stopping the blindness that's related to diabetic retinopathy. So we can stop people from ever developing DME and other complications. I think that's a game changer. I would think you would agree with that.
Every time I go over and I give talks on this, so I have opportunities to review, the data. Every time I review the panorama data, the Lucentis data that came out sort of incidental to DME treatment, the reversal of diabetic retinopathy remains the best thing that we do, but we don't do a lot of it because of the complexities of having those patients come in so often to get injected and so on and so forth. So yes, it's super exciting to have the potential to do that kind of reversal of disease with more durable treatments that can be applied less frequently. That I think that really would be a game changer in diabetic retinopathy, and I think most of my colleagues would agree. I want to talk a little bit about you personally in the company. We're friends. I know a lot of the things that you do, but you're pre-commercial now. You're a master of all these things: strategic planning and then commercialization. I guess what I would ask is, without knowing the crystal ball about when commercialization would be able to occur, assuming there's data to support that, what would be your role now as compared to, say, in a hypothetical post or during commercialization process, you individually?
Yeah, so the set of responsibilities I have today at our organization are from beyond retina. I'm responsible for all of the other franchises we operate in. And just to give you a little bit of background on REGENXBIO, the main core areas of our areas of expertise and what we call our capabilities are distributed across retina, neuromuscular, and CNS. We have a program in CNS today that we recently announced is going into pivotal program, and we intend to launch that product, addressing an unmet need in MPS II patients where these kids do not have an enzyme that keeps their brain function going. And over time, they don't develop the same neurocognitive skills as we do. So these kids ultimately grow up without the normal functioning neurocognitive function. So it's a debilitating, life-threatening disease. That program is moving forward now, and hopefully, we have announced a 5x25 strategy where by that point we will have programs like that, along with all the work we're doing on 314 being in pivotals today, along with the SCS program being in phase 2s, with wet AMD and diabetic retinopathy, all of these evolving to mature pivotal stage programs or potentially translating into commercial products. So we intend to have by 2025 at least 5 programs that are coming forward on the basis of maturity into phase 3 programs, and hopefully approved commercial products. And one of those big contributors there is the RGX-314 subretinal program in wet AMD. As you know, we're in the midst of recruitment, and we're on track to submit a BLA by 2024. So hopefully, we will have the data readouts happening as you think about backtracking from the date at the right appropriate times, and we can have a gene therapy available to patients as early as 2025. So that's the lead asset, and as I said, the MPS II program is on track to hopefully evolve to that mature commercial stage product by 2025, as well as all of the other stuff we're doing in neuromuscular and such, where we have a program in Duchenne muscular dystrophy, which is a huge unmet need, with almost 15,000 boys prevalent today in the US, where they lack the genetic material to produce a enzyme that keeps the muscle function going, called dystrophin. So we have a program that actually puts the transgene into these kids that can produce microdystrophin, which is the truncated form of the full protein that retains all of the functional elements of the protein. So I'm busy beyond retina at the organization. I'm also responsible for our pipeline portfolio efforts. We have a large organization that's focused on research and development with the type of capabilities we've built over the last 12 years as an organization across three very distinct therapeutic areas where there are a lot of unmet needs. So we are exploring all types of opportunities through our R&D group to see what type of programs we can bring forward beyond the programs we have in the clinic today. I also support all of our business functions, from corporate development through business development, through all of our planning efforts on a long range basis, to think about our financial needs, our strategic process to keep extracting value out of the technology we've built and the capabilities we've built. I think you know this, we've installed our own manufacturing facility, which is a GMP facility for gene therapy, which is a pretty big accomplishment in the context of the nascency that still exists, to have the ability to manufacture gene therapy products on a GMP basis. So we have built all of these internal capabilities, and my job on an everyday basis is to think about how do we leverage all of these to bring our products that are in late stage development and early stage development, and in our portfolio view, ultimately to as many patients as possible across these therapeutic areas.
When these products do get to commercialize, then the company will get to also lean on the 800 million personal relationships you've developed in a 25-year career, being well-liked and well-respected. That'll be brought to bear soon too. But you wear a lot of hats indeed, and I knew that, and I wanted to give you an opportunity to share some of that. And also now, you wear some hats outside of REGENXBIO. I want to give you a chance to talk a little bit about that. You have some other positions and board memberships. You're chairman of the board of directors at Avant-Garde Bio, a board member at Inovia, and a board member with Orbis, which was near and dear to all of the ophthalmologists' hearts. We feel good about what Orbis does and has done. I want to give you a chance to tell us a little bit. I'm particularly interested in Avant-Garde and their approach to gene therapy. How's that different or similar to what you might be doing at REGENX? And then maybe you can tell us a little about where Orbis is now. We're all familiar with it, but we don't stay in touch that much. It's great work that they're doing. Maybe you can share with us those two things.
Yeah, it's maybe I can start with Avant-Garde first. It's a company out of Italy, and it was founded by Alberto Arrucchio, who has contributed immensely to the world of gene therapy. He, in fact, was at University of in Philadelphia and worked with Gene Bennett during his training, and contributed heavily to the development of Luxturna as well. And he's a scientist that has been contributing in the gene therapy world beyond retina, and even in spaces like CNS and such. And the institution that he actually does all of his research is called the Telethon Institute out of Naples in Italy. That is considered to be one of the most premier gene therapy institutions in the world. So he has been on a quest to solve for large genes that cannot be packaged into current limitations that AAV has. If the current AAV can only package about 4 kb of genetic material. So if you have to actually express a protein that requires a larger gene than that, you have to find alternative solutions. So what Alberto has been doing is working on dual vector technologies where you can use that technology to solve for the limitations and that exist today in terms of packaging large transgenes into AAV. So the two technologies that he has discovered are based on being able to package two different transgenes into two different AAVs and bringing those transgenes together through a process of concatenation in vivo, or being able to produce proteins with two different transgenes that are being transduced into the cells through two different AAVs, and then on a post-translational basis, bringing those proteins together through the sticky ends and being able to express a full-length protein. Pretty cool science. So think about the second technology is actually based on how the unicellular organisms compensate for the ability to produce the proteins they need that are beyond their capacity to be packaged into unicellular organisms. So they produce these proteins with these sticky material called inteins that bring these proteins together, and then the inteins get excised. Then you have the full-length protein that you need. So we're applying that same concept in the human body now. So I got discovered by Sophie Nova through the process where they reached out to me and asked if I would be willing to be a part of the organization. And that process led to me connecting with Alberto. And Alberto had worked with Ken, who's our CEO back in the day, and also Olivier, who's our CSO back in the day. They confirmed, that it would be a good place for me to go and contribute and, help build an organization that seems to be solving for an issue that is today limited by the technology that exists with AAVs. And the way to solve for it is by these dual vector technologies and being able to produce the full length protein. And I'm very excited about the application of these two platforms in the context of 1,200 different mutations out there that require that full length protein where you can package given the payload limitations that current AAV has.
It's incredible. It's really complex science, but wonderful that you're participating, and credit, REGENXBIO for being flexible to allow you to, this is cross-pollination that the field needs. Honestly, your skill set can bring a number of these things, help to bring a number of these things to help patients and to be commercial products, and good for you and good for them. I'm really pleased to hear that you're doing this. What about Orbis? What are you doing with Orbis, and where are they today compared to 10 or 20 years ago?
And Orbis has been one of those contributions for me, having been in this space for almost 2 decades now, Firas, where, in fact, I got introduced to Orbis through a good friend of ours, Praveen Dugal, who is, currently the president of REGENXBIO. I've worked with Praveen for a very long time, and like you, he's a really good friend that talked to me about Orbis and the set of things they were doing. I've always observed Orbis from a external standpoint, and then I got involved with their scientific advisory board. All that translated into me getting to know the board of directors there when Praveen was one of the board of directors. And I just thought the organization's goals and what they were trying to solve for on a sustainable basis was something I would want to be part of and contribute to, both in terms of learning and impacting the organization to take that larger cause of solving blindness on a global basis. Orbis is a organization that's focused on finding sustainable ways to solve for blindness that exists around the world that's preventable. That's a bigger concept. Now they're also moving into a lot of the retinal diseases like diabetic disease, where we just talked about the preventable aspects of diabetes and the associated blindness, right? So a lot of things that were very similar, really connected with the board of directors there and the CEO and the type of things they're doing on a global basis. The institution has a flying eye hospital, and the intent there is to be able to go to remote areas of the world and use the flying eye hospital as a training institution to bring in people and literally put them through a short course of residency in ophthalmology and fellowships in ophthalmology, where they get trained on all of the right equipment, and then the organization builds the infrastructure, and that really translates into local sustainable care to prevent blindness. So it's with everything that I do on the corporate side and for profit, and my involvement with all of you in the community, this was my way of giving back in terms of time and effort. And as I said, Ken and the organization at REGENXBIO is very supportive of these things I do, and it's one of the things I really enjoy, working with my peers and the rest of the leadership, and Ken as the CEO and my manager, that gives me this flexibility and optionality to be able to contribute to the community beyond the things I do at REGENXBIO. So it's great.
It's phenomenal, and Orbis and you and others involved deserve a lot of credit. That's really one of the great philanthropic organizations I've known in my career, and I'm happy for you that you're participating in that. That's incredibly rewarding, and has been and will continue to help millions of people. It's fantastic. The training for other doctors is great. They're taking care of patients in remote areas, and now it sounds like they're also burgeoning into new biomedical areas, like say diabetic retinopathy. This will be great and very productive. Final point and question, more broadly speaking, I'll let you look in the crystal ball. Nobody knows retina innovation, the marketplace, better than you. Honestly, I mean that sincerely. Where's retina marketplace in 5 to 10 years? Are we still going to be leaders in innovation as we are now for the medical specialists in other fields as well? We take the lead. Will we be fixing everything with gene therapy by that point? Will we peter out? Where do you think we'll be 5 to 10 years from now?
I think every technology has limitations for us, and every technology has components of innovation that if you apply in the proper areas, you can really extract the best value. I think, from the introduction of, we talked about Macugen to Lucentis to Eylea to all the things that have happened in between with Beovu and now recently faricimab, and some of the innovations that we all expected to succeed in. You and I talked about the risk of drug development so many times and didn't come through, right, as part of large programs. And it's a disappointment, not just for shareholders, but for patients, right, and providers, where there's so much work that goes into these, and you just come on the other side hoping all of this is going to work, and we need to continue to do this, knowing those risks exist. And I think that's what makes drug development so special. And also the experiences you live with drug development. I think drug development makes you a better drug developer over time to be able to assess risks and take them forward. But no matter how good you get at it, you still leave that component of risk on the table that's unknown. I think when we look at the future, I'm so excited about what's happening with what we're doing, as well as what others are doing in this space, right? So some of the bispecific technologies that are evolving, and who knows tomorrow what faricimab has brought forward as a complementary mechanism could be built upon with gene therapies, right? It's and the contribution of that in the context of the type of outcomes you can accomplish, and what we're doing today in the concept of this single therapy with the optionality and flexibility you have. That's what I'm very excited about what we're doing. is with 314, this concept of foundational anti-VEGF therapy that can sustain the lifetime of the patient, I think can change how you manage these patients on a post-treatment basis, even in the context of future innovations, right? Because with gene therapy, you're leaving that optionality and flexibility with the clinicians, where tomorrow, if there is a complementary mechanism, and if a patient's already on 314, and you just have to give one more injection to enhance whatever outcomes you're looking for, now you have this foundational anti-VEGF already in place. So it's, I think, beyond this burden of treatment that we're trying to solve for. What's exciting for me about what we're trying to do here is that concept of being able to manage patients in the best way possible, even with taking into account the future optionality and flexibility for the patient. And the data is consistently pointing us to that when you do have this sustained expression of a protein versus the cyclic nature of what you do with intravitreal treatments of these peaks and troughs, there seems to be better outcomes. And it makes sense to me that even on a microscopic basis, you're probably treating the disease to a better extent, which you will realize over the long term that you're able to give the best outcomes to the patient. So I think all those concepts combined with the recent data that has come out of dry AMD is a great start for solving problems in dry AMD, right? I was hoping these therapies were going to read out in a way where they get introduced to the market and we're starting a option for patients and providers so we can keep building on it. I mean, that's where Macugen started, and now we're here with so many different optionalities to solve for the unmet need that has come through and keep giving the best possible solutions to providers, patients, and healthcare systems. So for me, it's also absolutely fascinating to see what's happening in the space of IRDs. I mean, now we're starting to think about not just monogenic diseases in the context of treating them early on, but even late stage monogenic diseases where you've already caused a lot of damage, where you can bring forward things like optogenetics, right? Because you're almost now trying to excite cells that don't exist anymore by going to cells that have survived beyond that photoreceptor degeneration and bipolar cells and such. So, I mean, we're living in the best era right now of all of the innovation. And I think retina is leading that in many ways across therapeutic areas. And for us, just to give you a little bit of background, the drug that got launched for spinal muscular atrophy called Zolgensma is changing patients' lives forever. And that drug has been introduced by Novartis, but it was originally our technology that was licensed to a company called Avexis that got acquired by Novartis. And now patients that have SMA are newly getting diagnosed on a newborn screening basis because there's a treatment that could help these patients preserve all of the function if it's administered early on. That really contributed to every state adopting now this newborn screening, where once patients are identified with this mutation, they can have access to Zolgensma. And that drug has been adopted now in many countries. And it's, as Novartis has reported this publicly, it's a $2 billion drug coming into 2022. These type of innovations are happening beyond retina too, with therapies like gene therapies. And we should continue to focus on what's right for patients, and whatever is right for patients is always right for providers, healthcare systems, and industry, from where I come from.
100% agree. It's remarkable what you just described in that other specialty. I didn't know about that. The advances have been amazing, and I think we're just at the beginning. Our specialty, our field is super lucky, and our patients are super lucky to have you with all your skill and energy focused on it. We're really fortunate to have people like you. I've known you for a long time, Ram Palanki, my dear friend. I thank you for coming on, and I thank you for being a part of this retina innovation era that I agree is one of the greatest in a long time, and it's continuing. Thank you for coming on.
Thank you, Firas. Always a pleasure talking to you, my friend.
Thanks for listening to the OIS podcast. Tune in next week as we chat science, medicine, and industry with ophthalmology's leading experts. Visit OIS.net for more information on podcasts, events, and exciting new features.